recent works here. So as a long-term research interest, my laboratory has been trying to understand the mechanism how mesenchymal stem cells are involved in tissue repair and remodeling. And as we know that bone marrow mesenchymal stem cells were initially recognized as skeletal stem cells that give rise to osteoblastic lineage of cells and contributes to bone formation during skeletal development and bone remodeling in adults. Now we know that mesenchymal stem cells exist in many tissue. But bone marrow is still considered as the main home of mesenchymal stem cells. So if we look at this diagram, this shows the localization of mesenchymal stem cells in bone marrow. And this is a piece of bone. We can see this is the osteoblast bone surface. This is bone marrow. The mesenchymal stem cells mainly resides at the outer space of this bone marrow sinusoids as a perivascular cells. Previously, because there's no single specific marker to label this population, and the MSC population were prospectively identified by using sets of cell surface markers. Here are at least 3 sets of surface markers that are commonly used to analyze or isolate MSCs from bone marrow. So my laboratory, this is the marker we commonly use to isolate this population from bone marrow. And in recent 5 years, actually, several single markers have been identified that can mark a subpopulation of MSCs in bone marrow. So, for example, in 2010, Paul Furness group from Albert Einstein Institute, what they found is the bone marrow cells expressing Nestin that have the ability to both self-renew and multi-differentiate, has a multi-differentiation potential. And also this Nestin positive cells in bone marrow constitute essential HSC niche component. So they think that the Nestin positive cells in bone marrow represent a subpopulation of MSCs. So like other stem cells, the MSCs have, as I said, has multi-differentiation potential. They give rise to a spectrum of mesenchymal cells, such as osteoblasts, adipocytes, and chondrocytes, myocytes, so on and so forth. So because of these features, MSCs are considered as injury drugstore. So whenever there is an injury, there is a tissue injury or any system has a disease in a disease condition, these MSCs can be released from their original niche, such as bone marrow, and mobilized to the blood circulation and migrate to the injured or diseased sites. So far, there are 2 fundamental questions that to be resolved in this field. One question is how can the MSCs mobilize or migrate to the target sites in response to tissue injury or in disease conditions? And another question is how can, if this mesenchymal stem cells can recruit it to the injured sites, how can they commit or differentiate into specific local cells to participate in tissue repair and remodeling? So, the goal of our studies try to, for long term, try to address these questions. So in this project, the model system we use is arterial injury model of mouse and rat. So the reason we use this as a model, because I consider myself as a bone biologist. I'm not a vascular biologist. So initially we use this model because we think that the artery has a very simple structure, and it contains mainly 3 layers. The innermost layer is intimal layer, mainly contains endothelial, just single layer of endothelial cells. And in the middle is called medial layers, made of smooth muscle cells. And the outermost layer is the adventitial layer, is actually connective tissue. So another reason we use this model is because after injury, actually is, in the mouse or rat model, there's if we do the endoluminal injury, that is the whole endothelial layer is damaged. So after that, almost every animal develop this new intima. This is pathological vascular remodeling. This is a new intima formation. So now it is clear that these new intima cells are alpha SMA positive smooth muscle-like cells or myofibroblast cells. So more importantly, this arterial injury model just mimic the clinical restenosis after coronary angioplasty. So if there is a blocked area for this coronary artery, usually the doctors put a catheter inside and with a balloon. And then once the balloon is inflated, so the lumen of the artery is opened up. And then the balloon can be removed and then left a stent there to keep the artery open, so the blood flow is restored. So one of the problem, the limitation of the treatment is in 6 months, about 30 to 50% of the patients develop this restenosis, which pathologically is the new intima tissue formation. During the past several decades, regarding the origins of the new intima tissues, there are intensive studies. However, the origin of the cells are still controversial. So classically, it is believed that this new intima cells are derived from the medial layer of vascular smooth muscle cells and adventitial fibroblasts. So these local vascular cells can migrate to the injured sites and then form new and proliferate to form new intima tissue. And another opinion is because there are resident vascular stem cells or progenitor cells, so these cells also involved can migrate to the target sites to contribute to new intima formation. And in recent 10 years, I would say, more and more evidence indicate that the circulating or stem cells or progenitor cells involved in this process. So initially, we try to confirm whether mesenchymal stem cells can migrate to the injured sites to contribute to new intima formation. So here we use the rat model of balloon insertion-induced injury in carotid arteries. So this is the sham control. We can see this is the 1 week after arterial injury. We can see that very thin layer of new intima is formed here. And at 2 weeks, that's the thick new intima tissue is formed. And then we detect whether MSCs can be detected at the injury sites. First we use Nestin as a marker. We can see in the sham control group, we did not detect any Nestin positive cells. However, at 1 week and 2 weeks after injury, we see a single layer of Nestin positive cells, which shows green here. So just a single layer at the intraluminal side of the new intima tissue. And we can see the deep layer. This is a new intima formation. The new intima tissue in the deeper layer, we cannot see Nestin expression. So we thought the Nestin positive cells, once recruited there, they differentiated into the myofibroblast or smooth muscle cell-like tissue, and then they lost the Nestin marker. So I will show you more evidence later. So actually, due to the time limit, I will not show you all the data. We use different MSC markers to detect. We got the quite similar result. And then we want to test whether these cells are derived from the migrated from circulating blood circulation. And then we detect the Nestin positive cells after arterial injury in mouse. And we found that at 3 days to 2 weeks, Nestin positive cells in blood significantly increased compared with sham control group. We also detect the MSCs using this 4 surface marker. So this MSC exhibit a similar pattern. So at 3 days to 2 weeks after injury, MSCs significantly increased in the blood circulation. And then we want to see whether these Nestin positive cells, whether once they recruited there, whether they contribute to new intima formation. So we use this lineage mapping mouse model. So by crossing the Nestin promoter-driven Cree mice with the ROSA 26 EYFP mice. So in the offspring of these mice, all the Nestin positive cells and their daughter cells are labeled by this EYFP fluorescence signal. So that means all the Nestin positive cell derived cells we can detect using the EYFP fluorescence marker. So we can see this is sham group and 1 week, 2 weeks after injury. So in sham group, consistent with previous result, we did not see any EYP positive cells. And at 1 week or 2 weeks, a lot of green cells we can see in the new intima tissue. And then we counted the EYP positive cells. About half of the new intima cells express EYP. That means half of the new intima cells are derived from Nestin positive cells. And then the next question we've asked is, what is the mechanism? What is the primary factor that induce the MSC mobilization, migration to the target sites? So this is what it is proposed in stem cell involved tissue remodeling. So once there is a tissue injury and the injury sites, the injury tissue will produce pro-migratory factors and then locally form a gradient to induce the migration of the cells to repair or remodeling the tissue. And on the other hand, the factors can also infuse into the blood circulation. The high concentration of these factors may induce the mobilization of the cells from their original niche to the blood circulation. So the question we asked is, what is the pro-migratory factors in this situation? Okay. So then we developed the ex vivo transvaal assay. What we did is we isolated the aorta from rat and then cut it open and make endoluminal injury. And then we cultured the uninjured vessel or the injured vessel. We collected the conditional medium. So, and then we placed the conditioned medium at the bottom layer of the bottom chamber of the transvaal and MSC on the upper chamber. And so the purpose is if there is any factors released from the injured vessel, so this will induce the cell migration. So as we expected, only the injured aorta conditioned medium induced the migration of the MSCs. Here is the quantitative data. And then, so I just cut the long story short. We detect a lot of factors in the conditioned medium, and finally we figure out is the TGF-beta-1 is the factor in the conditioned medium that induce the migration, because if we add the TGF-beta neutralizing antibody, there is almost a block the migration of MSCs. So this is another piece of in vivo data. We inject the mice with an inhibitor of TGF-beta receptor 1 and then totally block the Nestin positive cells recruitment at the new intima tissue. And then, so we also identify the downstream signaling molecules. We found that TGF-beta through activate SMAD signaling pathway and promote the production of MCP1, which is chemoattractant. Promote the production of MCP1 in the injured tissue, and the MCP1 can mediate the migration of MSCs. So this is we just inject the antagonist of MCP1 receptor and also block the recruitment of MSCs. So just summarize this part. So what we found is in response to arterial injury, TGF-beta is released, and then the active TGF-beta-1 in turn promote the production of MCP1 and induce the migration of the cells. So and the next question is, after the cells recruited there, do they differentiate into myofibroblasts to form new intima, or they differentiate into endothelial cells for repair? So again, we use this mouse model. What we found is this is the uninjured control, and the green cells, which is Nestin derived, positive cell derived cells, and we can see majority of the cells expressing alpha SMA. That means majority of the Nestin positive cells differentiated into new intima myofibroblast, but none of them differentiate into endothelial cells. And then we also did differentiation assay using the conditioned medium, which I mentioned previously. What we found is actually TGF-beta-1 also has multiple functions. And one function is induce migration, another is promote the differentiation of the cells into myofibroblast. And then we identified that we use different inhibitors of TGF-beta-1 TGF-beta-1 can stimulate different signaling pathways, and we find out that RhoA signaling pathway, is mediated TGF-beta induced MSC differentiation, and finally, we used an specific RhoA ROCK inhibitor called Fosdel, which is an FDA-approved drug. We treat the mice with Fosdel for 4 weeks. What we found, surprisingly, not only the new intima formation is inhibited, and the cells the nestin positive cells differentiated into endothelial cells. So that means the Rho A inhibitor treatment divert the differentiation of MSCs away from myofibroblast into endothelial cells for repair. So this kind of in vitro assay. So due to the time limit, I just summarize what we found is so the MSCs contribute to the neointima from the pathological arterial remodeling, and TGF-beta released from the injured sites can regulate two different action modes on MSCs. One is induce the migration of MSCs through SMAD MCP1 signaling pathway. Another function is to promote the cell's differentiation by inducing Rho A, by activating Rho A signaling pathway. So blocking Rho A ROC signaling pathway, so the MSCs differentiate into endothelial cells for repair. And right now we are trying to figure out the downstream molecular basis for why, so underlying this phenomenon. I just want to acknowledge all the people who contribute to this project. This the lab postdocs or graduate student, and this is our collaborators. Thank you.